Hi, hello everyone. So this is the first chapter of the second PU which I am making from a biotechnology, this principle and processes. First unit of the biotechnology and second is the application. So today we learned in this the meaning which is given by the European Federation of Biotechnology, how they are given. So before starting, I want to say that it's been being a time delayed for making the video. The day after of the 2 days, I will upload the videos. They will not be delayed for the next video. So let us begin this chapter. And after this, watching this video, do not miss to like and share with the video and comment down how you like the video and what are your queries. So let's begin. So the biotechnology principles and processes is derived from the two combination of words biology and technology. Usually there's a biotechnology. We can split in a two words, bio and technology. Bio means it's a biology in a life system, in an environmental system we have acquired and using with the technology. So the term was coined by the first scientist is Carl Ericke in 1917. The exact year is 1917. I think in the NCRT it's been given a wrong. So in 1917. Next point. So I said it's derived from the two combination of words which is biology and technology. So by combining these words they give the meaning. So it involves a group of techniques. Biotechnology involves a group of techniques that share a common characteristics of working with living cells and their molecules. Biology means it's life science, it's an environment which is from organisms can acquire some living cells and their molecules and making in a working in a common that we're not giving in a technique that is in a biotechnology. So in recent years this biotechnology has expanded in its scopes and applicabilities. It's now most relevant. So the definition in 1988, which is not given in NCRT, in the year of 1988, the EFB, European Federation of Biotechnology, it can be asked as an abbreviation question, EFB. So the European Federation of Biotechnology gave the definition of biotechnology by encompasses, by which encompasses both traditional view and modern molecular biotechnology. So traditional view and modern biotechnology is being a differentiation. So what's give difference means from a traditional, the old system of biotechnology, how they work, and from modern molecular biotechnology by new biotechnology. So comparing both, they gave the European Federation of Biotechnology give the definitions. So by encompasses means they have by differentiating, by comparing both traditional and molecular biotechnology, modern molecular biotechnology, they give a definition that the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. Means the integrating, they all are integrated of natural science, organisms, these cells, these parts of, and thereof, which in a molecular analogues, using by making a technologies, making for the products and service which is useful for the mankind. This is the meaning of biotechnology given NCRT important line. So here we are see the traditional view and modern molecular biotechnology. What is then those are first one coming to traditional, which means old biotechnology. So it is usually It is actually the kitchen technology. Kitchen. So usually we say in a house the main part is kitchen. So kitchen means it's like a primary in the house. So like this tradition also it is actually the kitchen technology developed by our ancestor using fermenting bacteria, and it is known that human beings have been using microorganisms for thousands of years. So here human beings are using the microorganisms for thousands of years to obtain the products like a curd, ghee, vinegar, alcoholic beverages, bread, etc. So without knowing the microorganisms were involved in this process. So in the old system, they are making from a thousand of years, but in a modern new technology we come, it can be make in a 6 months or 3 months. It's a fast process. So here the traditional biotechnology have a various applications. Areas of traditional biotechnology are: first one, fermentation to produce the foods. Fermentation to produce the foods is used to produce the products such as wine, beer, vinegar, bread, yogurt, etc. So by fermenting, so the first application is fermentation to produce the foods. It can be produced like wine, beer, vinegars, breads, and yogurt, and these processes are still in use today. We are not delaying this application. Still today we are using for the production of modern foods also. Industrial fermentation. So another application of this old biotechnology is industrial fermentation. In industries, the process of manufacturing like chemicals such as glycerol, butanol, like this preparing in industries, it's also development of industries process, which is an application of industrial fermentation. Third one is food preservation. So preserving means it's like storing. It's preserving, making preservation techniques like drying, salting, and freezing, which are used to prevent food from the contamination. That's a food preservation. Fourth one is selective plant breeding. It is a part of early crop improvement technology. It's a part of early crop environment improvement technology. It includes formation of new combinations. So in a selective plant breeding, which means we have to select. They are selecting. So it includes the formation of new combinations. Next, coming to the modern biotechnology. So modern means in a new form. So now the conventional industries, pharmaceutical industries, agro industries, etc., all industries are focusing their attention to produce a modern biotechnology based products. This is the most we have watching now also. By coming of the corona also, biotechnology has been improved, very improved, and have now very good scopes. So the two major features of technology which differentiate the modern and traditional biotechnology. So now I told the traditional biotechnology, old biotechnology, how they are using, what is the various applications of that. Now we have to compare the modern to the traditional. So there is two major features for this. They can differentiate the traditional and modern. First point is the capability of science to change the genetic material for getting the new products for specific requirement through recombinant DNA technology. There is one technique called as a RDT, recombinant DNA technologies, which is requirement for getting the new products for a specific requirement. for a new product and specific requirements, they have a capabilities of that changing the genetic material. Changes the genetic material. That process is recombinant technology. So the sentence can be seen that it is a fast working process. It's a fast working process which is in a modern biotechnology. That is a changing of genetic material, the capability of science that to change the genetic material for getting the new products. We can make it in a fast system for a specific requirement through the recombinant DNA technology, RDT. This is the first point. Second point is the ownership of technology and its socio-political impact. We have seen that the fast system has that who taken this the ownership of technology. They also been developed, and its socio-political impact. It not should be impact in a bad system, in a good way. It should be impact in a socio-political. So the second point is the ownership of technology and its socio-political impact. So the method associated with modern biotechnology is, so we say the capability of changing the genetic material. So the method associated in this with the modern biotechnology is genetic engineering, which involves the recombinant DNA technology and the gene cloning. Gene cloning means it's a duplicate. Cloning means getting a copies of parental, getting a copies of DNA or a vector, anyone. It means cloning. Cloning means it's getting a copies. So gene cloning, recombinant DNA technology. So the changing of genetic material, we have seen that one. So it's come under method of the modern biotechnology. So next the concept is genetic engineering and gene cloning. So next this is about the definition given by the EFP, but in the inside textbook they have given only the sentence, the first, the definition, the integration, which encompasses both traditional view and modern biotechnology. So I have give you the explanation both of the traditional and modern, which will be useful for your higher studies. So it can be asked in your NEET examination that traditional biotechnology, in which point of the view I don't know, but it can be possibilities to ask the question arises from the traditional and the modern technology. So in the next part we learned in a basis of the principle, so in the basis of like a question arising chapters. So here this and this here chapters, the definitions of the biotechnology on the, it is given by the European Federation of Biotechnology. So thanks for the watching this video. Like the video, share the video, and do not miss to comment down how you like. And next part, if you have any doubts, any queries, any other changes, you can comment down. Thanks for the watching.